Next is theory of biogenesis. It states that maggots, the larvae of housefly, are developed from the eggs of existing flies and not from decayed meat. It was also rejected by Louis Pasteur in 1864 and Sponzani in 1765. They proposed biogenesis. Next is cosmic theory. It states that the life came on Earth from some other planet in the form of seed or spore called cosmozoa or panspermia. These grew and evolved into the presently existing form on the fertile soil of Earth called panoply of living organisms, but it could not explain the mechanism by which panspermia survived adverse conditions, that is high temperature and lethal radiations of interplanetary space during its migration. It also does not explain the mode of origin of source panspermia and mode of its arrival to the earth. The next is catastrophic theory. It states that there had been several conditions such preceded by the catastrophe due to some major geological changes. Each catastrophe destroyed previous life completely and it was followed by more advanced creation. According to Orbigny, it's a scientist name, catastrophe has occurred 27 times. Each catastrophe destroyed the population as a whole and reproduction by recreation after each catastrophe. Other less known theories about the origin of life are as the theory of eternity of life and vital force theory. The next is chemogeny or the chemical evolution. Conditions on the primitive earth about 4 million years ago were such which favored the chemical evolution. When the surface temperature of earth was less than 100 degrees Celsius, the elements like nitrogen, carbon, hydrogen and oxygen were probably found present as simple inorganic or organic compounds, as their free existence was impossible at high temperature. So there many mechanism occurs, like formation of simple compounds, formation of simple organic compounds, formation of hydrocarbons, formation of oxyhydro derivatives of hydrocarbons, formation of fatty acids and glycerol, formation of complex organic compounds, formation of coacervates. So formation of simple organic compounds. As the Earth's surface cooled at 40 to 60 degrees Celsius, molecules and minerals present in water bodies combined and recombined in various ways through the process of condensation, polymerization, and oxido-reduction to form a simple organic compounds like alcohol, aldehydes, glycerol, fatty acids, purines, pyrimidines, simple sugars. Next is formation of hydrocarbons by by combining highly reactive groups uh, like acetyl, ethylene, methane, acetylene, and formation of hydroxy derivatives of hydrocarbons like aldehydes, ketones, and acids. Its examples are acetaldehydes, formic acid, formation of fatty acids and glycerols. By this process, the condensation of polymerization and of aldehydes and ketones by their oxidation. Formation of complex organic compounds. Simple organic compounds showed chance chemical reactions and polymerization to finally form complex organic compounds like polysaccharides like starch, cellulose, glycogen, etc. So formation of coacervates. Oparin and Sidney Fox proposed that complex organic compounds synthesized abiogenetically on the primitive Earth later tended to accumulate due to intermolecular attraction and formed large colloidal cell-like aggregate like coacervates. So here is the summary of steps in origin of life. These are free atoms. First of all, free atoms. Secondly comes inorganic molecules, and then comes simple organic molecules, then come complex organic molecules, coacervates, eobionts, free game, free genes. It's written as GEME, but it's GENE. So free genes, first primitive cells. So this is the step of the origin of life. Free atoms like hydrogen, carbon, nitrogen, then inorganic like H2, H2O, NH3, simple organic molecules like CH4, H2O, complex organic molecules like sugar plus sugar, fatty acids plus glycerols, sugar plus sugar equals to polysaccharides, fatty acids plus glucose is equal to fats, coacervates, aggregates of large complex molecules, organic molecules capable of growth and division. Eubionts are nucleic acid controlled coacervates. Free genes are self reproducing nucleoprotein combinations. First primitive cell is lipid protein membrane bound units with gene joined into long filaments. So, thank you.